Microbial interactions. So here we can see, as the name indicates, these are the microbial interactions. These are the interactions between the microbial population in which they establish their association between each other, within each other, and these associations could be positive or negative on the basis of the benefit or the harm they are getting from each other. Now, these are the ubiquitous, diverse, critically important in the function of any biological community. History, this was first, the term was given 1879, that was symbiosis. After that, Hamilton in 1964 gave the indirect interaction. Then Odom divided into the two groups, positive and negative. Now the microbial interaction, there are different types of microbial interactions. These could be interspecific and intraspecific. So interspecific means within between two species, and interspecific means when the intraspecific when the it is within the species. So interspecific interactions will be positive or negative. So these positive will be cooperation and competition, and intra-specific interaction could be neutral, positive, and negative. Each and every interaction we will discuss in detail in the coming slides. On the basis of the positive and the negative outcome, these interactions, some of the example of the positive interactions are mutualism, commensalism, and proto-cooperation. Coming to the negative interaction, these are amensalism, parasitism, predation, cannibalism, and competition. So all these type of interactions coming under the negative interactions. So let's start with the positive interaction. Firstly, the first interaction is the mutualism, which is under the positive interaction. We all know that mutualism, it is a term that describes the relationship that is the mutually benefited. So partners which are there involved, these are the mutually benefited partner. So it is in the way here the this is also called as the cooperation. It is highly specific and obligatory interaction. But there is one more term, that is the symbiosis. So sometimes it is a confusion between the symbiosis and the mutualism. So let's discuss it here only. That symbiosis, that means living together. It is an ecological interaction between at least two species where there is persistent contact between the partner. Mutualism here, at least two species, when both the partners are the benefited from this relationship. Okay, that means all the mutual relationships are symbiotic, but all symbiotic are not mutual. Okay, so all symbiotic are mutual relationship, but not all the symbiotic relationship are mutual. Means all the mutual in which, because they have to live together and they are getting benefit, so that will come under the symbiosis. Coming to the example of the mutualism. So mutualism, here we have taken five examples. First is the rhizobium legume association. Second is the mycorrhiza. Third is lichen. Fourth is herbivore microbe interaction, and clownfish and sea anemone. So firstly, we will see the rhizobia legume association. We know that rhizobium is a nitrogen fixing plant, and it is present and attached to the root nodules or root of the leguminous plant. So once they attach, they will enter into the this plant, an infection thread will be formed through which the bacteria will enter the root cell, and then they will form the bacteroid. So after the bacteroid formation, the enlarged root cell will form the nodule. Now we have seen this very familiar, we are familiar with this structure in the leguminous plant. These nodules are present. Now there is a question, what is happening inside the nodules? So this inside the nodules, the bacteria is converting the with this nitrogen that is present in the atmosphere into ammonia, and which can be used by the plant. Okay, so that is the role of rhizobium in the leguminous plant. Coming to the next example is the mycorrhiza. Again, this mycorrhiza is the association or a relationship between the root system of higher plants and the fungus of the fungal hyphae. So higher plants and the fungal hyphae. This is the mycorrhiza. Now, these mycorrhiza are also, on the basis of their structure, on the basis of their presence, on the basis of their approach inside the higher plant, they are classified into three types: ectomycorrhiza, endomycorrhiza, ectendomycorrhiza. So let's discuss. First is the ectomycorrhiza. These are the common present on the forestry like pines, beeches. So what is happening here? Only main thing is this: these are the higher plant, and the fungal hypha. They are present like a sheath over the outer side of the root. So over, and they will form a mantle. So that is a covering that is formed, and this is also called as hearting net. This hyphal network form the heartig net, and what they will do, then they will extend inside the few layers of the cortex. They are not until the endodermis, only till endodermis, they don't enter inside. So these are called as ectomycorrhiza. These are the example, and this is the significance of the ectomycorrhiza. Coming to the endomycorrhiza, as the name indicates, here what will happen, this hypha, they will invade the root cell, and without forming, in this no mental formation is there, just they will enter into inside the root cells. Okay, now endomycorrhiza are of three types: vesicular arbuscular, orchidaceous, ericaceous. So orchidaceous and ericaceous, that is the name, depending on the families of the plant on which these fungal hyphae are formed. Now we will see it in detail also. Here you can see. So first is the vesicular arbuscular mycorrhiza, and this arbuscular means arbuscule is formed. So these are the most popular endomycorrhiza. It is a kind of mycorrhiza in which whose hyphae penetrate the cell of the plant root by producing balloon-like structure or a vesicle. Okay, and dichotomous invagination. So this arbuscule and the vesicles, these are the two structure which are present in the vam. So these are present in the roots of angiosperm, and the fungus involved here are the Phycomycetes fungus. Second is the ericaceous. So in the ericaceous mycorrhiza, in this type of mycorrhiza, the loose weft of hyphae is present. So loosely arranged hyphae are present here, and they will be and they will also penetrate the outer cortex. Okay, and these are present in the Ericaceae family. Coming to the next is the orchidaceous mycorrhiza. That is another type of endomycorrhiza in which the this is intracellular penetration of inner cortex is again here, and there is coil-like structure, that is pelton. So pelton is the characteristic feature of the orchidaceous mycorrhiza, in which the fungus will interact with the family roots of the orchidaceous family. Now this is the difference we can see. And endome, these are penetrating inside, and here they don't penetrate in the ectomycorrhiza. One more is the ectendomycorrhiza. These have the characteristic of both ecto and endomycorrhiza. So here the fungal partner establish the mantle of the hypha as well as the hyphal coil and hostoria, which will invade cortical cell of the root. So here what is happening, mantle is also formed, and with the help of hostoria and the coil, they will invade the cortical cell. This is the example of the ectendomycorrhiza. Coming to another type of mutualism is lichens. So in this, what is happening, lichen, we know that these are the complex life forms. Why they are complex? Because they are not a single organism. They are the combination of algae and fungus. So algae involved here are of the green algae and the fungal or the blue green algae. Both type of algae are involved. But the fungus, these are here involved are the Phycomycetes and the this Basidiomycetes and the Ascomycetes. Now, what is the function of algae and fungus here? In this type of interaction, alga is responsible for preparing the food, and fungus is the main component of lichen. They absorb water from the surrounding, protection and shelter to the alga. So, this is the main role of the this type of mutual relationship. Here you can see, if you go for the structure of lichen in this, the inner part, that is the cortical part, is composed of the compact hypha. After that, the algal zone is present, and then medulla zone, it is formed of loosely arranged hyphal structure. On the basis of their structure, they are of four types: crustose, scamulose, foliose, and fruticose. So crustose, as we can see, these are the thin, flat, no distinct lobes are present. They are attached to rocks, trees, and tree trunks. Coming to squamulose. Squamulose means here minute overlapping scales are present. So these are also called as scamules. So on the basis of the name squamulose, like a name is given. Coming to the folios. Folios, here it is a flat bread like structure, and flat broad structure. These are leaf like shape. So these are flat, these are broad, leaf like shape is there. Lower and upper lobes are present. They are present on the rocks. Fruticose, again these are the small and they are branched. So this was the different type of lichens. Two other type of mutualism examples are herbivore microbe interaction. So herbivore microbe interaction in the rumen of many herbivorous in the many this ruminants, the many type of bacteria are present. So what is the role of these bacteria here? What the cattle for the digestion of cellulose, these bacteria are present. So these bacteria get their nutrition from the food, and in return they will degrade the cellulose. Like cattle, they feed on the grass, paper, so all these have the cellulose. So that will be degraded by these bacteria. In return they will get their food. So that is one of the interaction. Then other mutualism example is the clownfish sea anemone interaction. So here we can see this is the clownfish sea anemone. In this, clownfish uses anemone for their interaction, for its protection from the predators. So in this interaction, clownfish is using these sea anemone for its production from the protection from the predators. And in return, this clownfish clean the anemone of their parasite and provide a source of nitrogen for growth. Now, coming to the second example, that is the commensalism. Second is the positive interaction, type of positive interaction. So, it represents a relationship between two microbial population in which one is benefited and other will remain unaffected. So, it is neither harmed nor benefited. It is unidirectional relationship between the species. So, here we can see that Mycobacterium leprae, once the infection is there, from the open wound, it will, from the open lesion, it will cause the those lesions present on the skin, and that will on the body surface they will allow other pathogen to establish the secondary infection. So once the infection of leprae is established, the other organism or the other pathogens which are coming, they are getting their they are benefited, but Mycobacterium leprae is not is unaffected. So this is the example of commensalism. Now coming to the proto-cooperation or the synergism. So in this association between two microbial population in which both the population benefit from each other, it allow the population to perform their metabolic activities involving the exchange of nutrients. So that is why this phenomena is called as syntropism. Means both are benefited and both allow the exchange of the nutrients. So this way, this is the very interesting type of interaction, that is proto-cooperation or synergism. So here example is desulfo vibrio bacteria, which supply the hydrogen sulfide and carbon dioxide to chlorobium bacteria, and in return the chlorobium makes sulfate and organic material available to desulfo vibrio. So that means that is the thus the mixture of two population produce more cellular material than either or alone. So when both of them are together, they are more beneficial to each other. After the positive interaction, we will come to the negative interaction. So first type of negative interaction is the amensalism. In this, a type of interaction in which one microorganism releases a specific compound which has negative effect on the other. So like we know the antibiotics, so what will happen when antibiotic was secreted, the bacterial growth was prohibited. Okay, so this is the example of amensalism. One more thing is ant use antibiotic producing streptomyces to control the fungal parasite in their fungal garden. So this is the, here you can see that ant, they use the antibiotic, that is antibiotic producing streptomyces to control the fungal parasite. This is the name of fungal parasite. Okay, so this will secrete antibiotic which will control the fungal parasite. So this is one type of the association. Now coming to the second negative association is the parasitism. It represents the relationship between the two organisms, and only one is advantageous and second will be harmful. For second it is destructive or uh, harmful. So this is also one type of mycoparasitism. Now coming to predation, it occurs when the microbes, the predator engulf and digest the other microbe. Okay, so predator will engulf the prey. So for this, the example is Dallovibrio bacteria and the other small gram-negative bacteria. So what is happening here? Dallovibrio is a wide spread in the aquatic habitats. So and attack the other bacteria by boring holes in the wall. So what they will do? They are boring holes in the wall, and they will cause the lysis of the cell and eventual release of many small vibrio-shaped bacteria. So this is called the predation. So here you can see this is the Dalobacteria host, and it is this is the Dalobacteria on the host. It is boring the cell wall, and it then will make the different more number of copies after lysis. They will come out, and this is the predation. Now, cannibalism is the act of consuming another individual of the same species as a food. So examples are human and snakes. Now coming to competition, it was also very important microbial interaction. It was studied in 1934, and it represents a negative interaction. So in here, both the population are affected with respect to their survival and growth. So in this, they share the limiting resources like space, limiting nutrients while surviving at the lower population level. So for that limiting resource, they are competing, and for the same resource. Okay, so example is E. coli and coliform. So they are competing for the food source, and azotobacter and cellulomonas. So they will compete for the, here you can see the glucose, this use of this azotobacter, they will use the glucose that is provided by the, this glucose will be utilized by the azotobacter, and these cellulomonas, they will use the nitrogen, and that will be, that has been fixed into the ammonium form by the this azotobacter name. Whatever nitrogen has been fixed by the azotobacter, that will be utilized by the cellulomonas. So both of them, they are using the uh, resources, and this is called the common this is called the competition. Now we can see, we can summarize it here with the positive and the negative. In the positive interaction, mutualism, if the two species A and B are involved here, in this both are positive, both are benefited. Commensalism mein one is benefited, other is not affected. Proto cooperation, both are positively affected, beneficial to both of them. Now amensalism mein one is unaffected, other is negatively affected. Then parasitism, again one is positive, second is negative. You can understand the positive means beneficial and negative means that is the harmful to that one. Predation, positive and negative. Cannibalism, positive, here one is consuming other. So first one species is positive and other is in the uh, for that is harmful. Competition mein both are negative. So these are the negative interaction, these are the positive interaction. In this we can see that we have studied all the type of interactions, microbial interactions. So we can summarize it that microorganisms, they engage in a wide variety of the social interactions. So many are there. Role of microbes in the plant growth, nutrient availability, so they are also demonstrated. Now there are increasing resistance in the research, and for that microbial interaction between the medicinal plant and their rhizosphere. So these all are the significance of the microbial interaction. So in this we have seen, in this presentation we have seen all the different type of microbial interactions and how they are significant for each other and what are their roles, and with a small details on their structural uh, structures also. Okay, thank you so much.